Good morning, grade 7 students. I hope you are doing well. Today I'm going to continue this activity, respiratory gaseous exchange. Please open your books page 49. In the previous video, we said that respiration or breathing is the process by which oxygen is absorbed and carbon dioxide is released in air or in water. All living things respire. All living things take oxygen and release carbon dioxide. How can we identify the gases involved in respiration? There are many materials, however, we can use these two, the oximeter and the lime water. To test that living things are absorbing oxygen, I use the oximeter. It is a device that measures the quantity of oxygen. And to test that living things are releasing carbon dioxide, I can use the lime water. Lime water is a colorless reagent which becomes turbid in the presence of carbon dioxide. Today we are going to conduct another experiment, however this time it is on plants, mainly germinating seeds. Let's read the experimental procedure. Introduce lime water into the reservoir of two containers A and B. Please check the picture. Place on a sieve germinating seeds only in container A. So in A we have germinating seeds, however container B lacks germinating seeds. So what is the variable factor in this experiment? Presence or absence of germinating seeds. Close well the two containers to prevent the leakage of the gases. Observe the results in a few minutes represented in this document D. Then follow the evolution of the oxygen in the containers with a digital oximeter. Pause this video for a minute and try to analyze this experiment. In container A, having germinating seeds, the lime water is turbid, indicating the presence of carbon dioxide, right? While in container B, lacking germinating seeds, the lime water remains clear, indicating the absence of carbon dioxide. What can we conclude here? What makes lime water turbid? It is true, germinating seeds. So, germinating seeds release carbon dioxide. If germinating seeds are releasing carbon dioxide, do they absorb oxygen? To determine whether they absorb oxygen or not, we placed an oximeter, a digital oximeter, and the results are represented in this document, document E. When you want to analyze the graph, start writing your analysis at time 0 minute. It is important to mention the quantity of oxygen at the beginning of the experiment and comparing it at the end of the experiment. What happens to the quantity of oxygen in both experiments? Use increase, decrease, or remain the same? I will answer. At the beginning of the experiment, the quantity of oxygen in both cases, the control experiment and the enclosure with seeds, is 21%. After 10 minutes, because these two curves end at time 10, so after 10 minutes, the quantity of oxygen in the control experiment, which is the experiment without seeds, remains the same, 21%. This means that oxygen here is not absorbed, because this container or this experiment doesn't have seeds, while the quantity of oxygen in the experiment with seeds decreases to 19.6%. What do you conclude here? That the seeds are absorbing oxygen. Okay, activity 1 is finished. Please, I want you to write all these questions on your copybook and try to solve them on a double sheet. Thank you and see you.